This is topic 4 e-lecture on biological hazards. In the first part of the e-lecture, we are going to look at the first 3 learning outcomes. That means listing out the different types of biological hazards and describe how laboratory acquired infections can occur and their main routes of transmission, and describe risk control measures for biological hazards. So what are biological hazards? Biological hazards are biological agents which has a biological origin, and they can pose a threat to the health of living organisms like humans, animals, etc. And this is the universal biohazard symbol. You should have seen this in different, let's say, containers of biological agents, hospitals, laboratory doors, which they handle biohazards, and biohazard waste bins, etc. This slide shows there are different types of biological hazards. So, example, blood-borne pathogens. The blood-borne pathogens are microorganisms such as bacteria, viruses, or parasites, which are carried in blood or body fluids. Example, hepatitis B and C viruses, HIV. These can cause diseases in people. And cell cultures. Example, we use human cell models or sometimes rodent cell models to conduct research to test the effect of certain drugs on cells. So these can be biological hazards as well. And we have genetically modified organisms, biological agents and toxins. Means certain toxins can be produced and extract from microorganisms like botulinum toxins. Viral vectors are viruses which we can use to carry genetic materials into other types of cells. So example is adenovirus. These are used in recombinant DNA technologies. Then we have parasites like protozoa. Zoonotic agents are agents which can carry diseases between animals to humans or humans to animals. Then example is influenza A, which is responsible for swine flu. And there are infected animals or insect vectors, and also allergens. Example, if people are working with laboratory animals, if they are consistently exposed to the animals, then the urine or saliva, this can trigger allergic reactions. So all these are different types of biological hazards. You don't have to actually necessarily remember the examples. Examples are given for your information and for you to understand the concepts. Laboratory acquired infections, in short, we call them LAI. They are categorized as all infections acquired when you're working in a laboratory or doing laboratory related activities, regardless whether they are asymptomatic or symptomatic in nature. So the laboratory acquired infections can occur via 4 main routes. We call them routes of exposure. The 4 routes are mucous membrane exposure, ingestion, inhalation, and percutaneous inoculation. Let's look at the details in the next slide. Let's look at some of the ways the laboratory acquired infections can occur via the 4 routes. So mucous membrane exposure can occur if the infectious materials get in contact with your eyes, nose, or mouth. Example, if you had contaminated gloves and you touched your eyes, okay, then the infectious material can get into your eyes. That's mucous membrane exposure. Or let's say you were handling some biological agents and you were not wearing safety goggles and some of them splashed into your eyes. That is also mucous membrane exposure. Okay, so that's via eyes, nose, or mouth. Inhalation, it's easy to understand. It's the inhalation of infectious aerosols. Aerosols are like tiny droplets which you can't really see from your naked eyes, but they can deposit in your hands while you're working with biological agents, or you can inhale them as well. Ingestion is via eating or drinking. So let's say if someone was working in the lab and didn't wash their hands properly before leaving the lab, and if the hands were contaminated, if you proceeded to eat or drink, then these hazardous agents can get into your system. Okay, that is via ingestion. Percutaneous inoculation means if you have accidental needle pricks which are contaminated with biological agents, or if you get cut with contaminated sharps or animal bites, or if you had, let's say, open wounds, broken skin in your arms, and the biological hazards can get into your system. This is via the percutaneous inoculation route. In the next few slides, let's look at some of the risk control measures that we can take in order to prevent or minimize exposure to biological hazards. So before we can look at risk control measures, we need to identify some things. One is risk group classification system. The other one is biosafety levels. Um, we need to understand that the biological agents are classified into different risk groups according to their hazard levels. So the international classification system is by WHO, the World Health Organization. The WHO classification system is they classify the biological agents into 4 risk groups. Example, risk group 1 organisms, risk group 2 organisms, risk group 3 organisms, and risk group 4 organisms. Okay, risk group 1 organisms are the least hazardous, and risk group 4 organisms are the most hazardous, most dangerous. Okay, and there are also like other classification systems based on different countries. Example, Singapore, we have Singapore Biological Agents and Toxins Act. So in Singapore, when we're handling biological agents, we need to refer to this, okay, in order to see what kind of preventive measures should we take to handle biological hazards. So in the Singapore Biological Agents and Toxins Act, they classify biological agents and toxins into different 5 schedules. Example, first schedule organisms, second schedule biological organisms, etc. So when we take risk control measures, we actually need to consider the risk group of the organism. This slide shows you the 4 risk groups by the WHO International Classification System and the corresponding Singapore classification, BATA schedules. BATA stands for Biological Agents and Toxins Act, and the description of those agents and some of the examples. So example, risk group 1, as I mentioned earlier, is the least hazardous, like E. coli. Okay, risk group 4, example is Ebola virus, which is a very, very dangerous biological agent. So you don't have to memorize the examples or the description. This is given for your information. The next point we need to consider when we implement risk control measures for biological agents is the laboratory biosafety level. Laboratory biosafety level, we call it BSL, okay, biosafety level. It is a classification system for laboratory facilities. So the purpose of this classification is they will tell you how to safely handle a particular biological agent. Example, let's say if you want to handle the SARS coronavirus, okay, then they will tell you which type of biosafety level should you use. Is it BSL 1, 2, 3, or 4? And what are the laboratory practices you should follow so that you won't get infected? And what kind of safety equipment or PPE should you use, etc. So these are based on a set of conditions required to safely handle a particular biological agent. So there are 4 biosafety levels: BSL-1, BSL-2, BSL-3, and BSL-4. BSL-4 is the facility where we have to use to handle the most dangerous or hazardous biological agents like risk group 4 organisms like Ebola viruses. BSL-1 is kind of like our teaching facilities, because in our teaching facilities we don't use hazardous biological agents. We use quite safe microbiological strains like non-pathogenic E. coli. Okay, then we can use our general PPE, right? We won't get infected. So these are like BSL-1 facilities. So this slide shows you the 4 biosafety levels and what kind of agents are handled in these biosafety levels. Example, if it is BSL-1, we handle known biological agents which are unlikely to result in diseases in healthy individuals. Example, non-pathogenic E. coli. Okay, BSL-3, we handle biological agents that may cause serious or potentially lethal diseases. So example is Mycobacterium tuberculosis, etc. This slide is to recap the hierarchy of controls in the risk control measures. I'm sure you have seen this in your topic 2 and topic 3 as well. Okay, so in the hierarchy of controls, elimination is the most effective risk control measure. That means if we can totally remove the hazard. Okay, sometimes it might not be possible actually. Substitution, that means can we replace the hazard with a low risk one? Engineering controls are physical means that can limit the hazard, like example use of fume hood when you use hazardous chemicals, or use of biological safety cabinet when you use biological agents. These are engineering controls. Administrative control means providing training to people. That means if you have like new employees, you can provide training to these people, okay, and you can have standard operating procedures like adherence to procedures, conduct safety audits. So all these are administrative controls. PPE is considered the least effective risk control measure in the hierarchy of controls. I think I have explained this in topic 2 earlier, because PPE actually does not contain the hazard, does not remove the hazard. It can only minimize or your exposure to a certain level when you are using the correct PPE. So let's see what are some of the risk control measures we can take for biological hazards. So when we implement risk control measures, we also need to consider the type of control in the hierarchy of controls. Okay, so elimination is the most effective if you can totally remove the biological hazard. So I have put if possible within brackets. Sometimes this may not be possible. Okay, and substitution, can you replace the biological hazard with a lower risk one? Let's say in terms of research activities, if you can use a lower risk biological agent, okay, then it would be better. This is substitution. Engineering control, remember earlier I mentioned it is implementing physical means that limit the hazard exposure. So example of risk control measures for biological hazards in terms of engineering controls is use of the biosafety cabinet, or BSC. Okay, you use BSC when you're handling biological agents which has the potential to generate aerosols. Okay, so BSC is a safety equipment used in BSL-2 facilities and above. That means in BSL-3, BSL-4, etc. Okay, this is an engineering control for biological hazards. Administrative controls, there are few. Okay, that means adherence to procedures and training to reduce or eliminate hazard exposure. Example, you can provide training to people before they start work. And you can have standard operating procedures in place. So standard operating procedures you can have for work procedures. Example, if you have to inoculate a certain bacterial culture, what is the standard operating procedure to follow? What is the standard operating procedure to follow when you want to dispose biological waste, etc. Okay, and some facilities or companies may have occupational health programs. That means, let's say in a certain laboratory, you have people who need to work with very hazardous biological agents. You can have an occupational health program for these people. That means you can actually provide vaccines or immunization for the agent that they are going to handle. And you can conduct routine medical checkups to see whether they have any infections or whether they have any exposure to this kind of biological agents that they are handling. So this is what I mean by occupational health programs. And we can also conduct safety audits and inspections to see whether these laboratories follow the safety protocols and rules and regulations properly, so that this will actually protect the people working in these workplaces. Okay, so all these are actually administrative controls. PPE, we know the type of PPE actually depends on the nature of the biological hazard that you are exposed to, like whether it's a risk group 1 organism, risk group 4 organisms, right? And whether you are supposed to handle it in what type of biosafety level. Example, in BSL-1 facility, we can actually use our general PPE. But if you need to use BSL-4 facility, right, you need to use a full body suit like positive pressure suit, because general PPE is not suitable to work in BSL-4 facilities. In part 2 of the topic 4 e-lecture, we're going to look at the last few learning objectives, which describes proper handling of biological hazards. So proper handling actually includes how do you handle particular biological agents using biosafety cabinets? What are the storage requirements for biological agents? And how do you actually dispose waste generated from biological agents? When we're handling biological hazards or biological agents, we actually need to know what kind of agent that we are working with. That means if your biological agent that you're working with are infectious and has the increased risk of airborne infections and also has the potential to produce aerosols when you're handling it, then this kind of biological agents must be handled inside the biosafety cabinet. In short, we call it BSC, and these organisms should not be handled on open laboratory benches. Otherwise, there won't be any protection to the user. Okay, so biosafety cabinet or BSC is actually a safety equipment used in facilities of biosafety level 2 and above. 2 and above means BSL 2, BSL 3, and BSL 4. Okay, to handle hazardous biological agents. So actually there are different types of biosafety cabinets like class 1, class 2, class 3, etc. Class 2 is the most common type that we actually see in laboratories. Class 3 is a completely enclosed biosafety cabinet, which are commonly used in BSL-4 facilities. So biosafety cabinets are enclosed, ventilated laboratory workspaces, and they are designed to contain infectious aerosols and splashes within the cabinet. Okay, so these BSCs are installed with filters called HEPA filters. These filters will actually filter the air which is drawn through the front grille of the biosafety cabinet, and that air is actually circulated within the cabinet. So biosafety cabinet is designed to protect the user or the operator from getting exposed to hazardous biological agents, and your work materials are protected too. They won't get contaminated, and it can protect the environment as well. The next few slides describes the operation of a biosafety cabinet. So in this slide, you will see the steps that you have to follow for the BSC startup procedure. So startup procedure means how should you turn on the biosafety cabinet? How should you bring in the materials? How should you arrange them in the biosafety cabinet in order for you to conduct your experiments? So I suggest you watch this YouTube video. I have already narrowed down the scope. You can watch from this timing in the video, which actually shows you the startup procedure, and you will be able to understand these steps a lot more clearer when you watch the video. Okay, so the first thing, you have to make sure that you wear appropriate PPE, personal protective equipment, suitable for your lab procedure. And in the biosafety cabinet, if you see that the UV light is on, you need to turn it off before you open the sash of the BSC. So usually UV light is used to disinfect the work surface of the biosafety cabinet when it is not in use. Okay, so before you open the sash, if it is on, you need to turn it off because you shouldn't get exposed to UV light. It is harmful. Okay, then you can open the sash of the BSC to the appropriate height. Okay, that means when you're seated on a stool or chair, roughly your operator's shoulder should level with the bottom of the sash. This is actually to ensure you can work comfortably, put your arms in and work, okay, not your whole upper body in. Then you can turn on the fluorescent light and the BSC blower. So it is very important that you turn on the blower or the fan, okay, because that's when the air circulation will start to occur. Okay, that's when the air gets filtered and the infectious aerosols can be contained within the cabinet. Okay, so it's very important that you turn on the blower or the fan and allow it to purge for a few minutes to remove any remaining contaminants in the work surfaces and to stabilize the airflow. All right. After that, you can actually disinfect the work surface of the BSC with a suitable disinfectant like 70% ethanol. Then you need to gather all the materials that you need to bring in to the BSC and also disinfect them with a disinfectant like 70% ethanol. Okay. Then when you bring in the materials to the BSC, you have to make sure that you only bring in necessary items. That means don't bring unnecessary items and overcrowd the biosafety cabinet. Okay. You also have to take note that when you're placing materials in the BSC, you should not cover the front air grill or the back grill or the rear grills, because the air circulation happens between these front grill and the rear grill. Okay, so if you keep items covering these air grills, the airflow pattern is actually compromised. Then if the airflow is not happening properly, it will actually lead to contamination of your samples and also affect the protection of the user as well. Okay, then when you arrange the work materials in the BSC, you also have to arrange it in three areas, like your clean items in one area. Usually your working area is in the middle, and should be a contaminated area where you can keep your biohazard bag, all the used items like pipettes, etc. Okay, so the general rule is the contaminated area should be at the operator's dominant hand side. Okay, because your workflow should be from clean to dirty area in order to avoid any cross contamination while you are working. All right. So after you load all the materials, arrange them into clean, dirty, and working area, you can leave the airflow to stabilize for a few minutes before you actually start your experimental procedures. Okay, so I strongly suggest you watch the video. Okay, as I mentioned earlier, so these steps will make a lot more sense to you when you watch the video. And you also need to understand there are factors to consider while you're working in a biosafety cabinet. So as I mentioned earlier, the sash of the BSC should be only open to the appropriate height. Okay, if you open it all the way up and put your whole upper body in, then you won't be protected from infectious aerosols, etc. Okay, then the materials which you bring into the biosafety cabinet should be kept to a minimum. Okay, you should avoid clutter, because if you have too many items in the BSC, it may also cause spillages. Okay, and you don't have much room to work. And as I said earlier, the front grills and the rear grills should not be blocked with items, because it will actually disrupt the airflow patterns in the BSC. Okay, this can cause the contamination of your samples and exposure of the operator to the infectious aerosols. Okay. Then when you're working in a BSC, you shouldn't have excessive movements of hands and arms through the front opening, and should avoid rapid side-to-side movements, because this can also disrupt the airflow patterns of the BSC. Okay, and multiple users should not use the BSC at the same time. That means usually we recommend only one person work in the biosafety cabinet at one time. If you have multiple users, that means more hand movements and also may lead to cross contamination between samples. Okay, and you should not bring paperwork. That means, like, your protocols, printed protocols inside the BSC, because if you place them inside the biosafety cabinet, right? Uh, During the work procedures, the droplets of biological agents or aerosols can deposit on your paperwork without you seeing that. Okay, then after your procedures, if you take them out and keep it on your laboratory benches, or if you bring it home, okay, that means you're actually spreading the infections beyond the BSC and also beyond the laboratory environment as well. Okay, so you should not bring paperwork inside the BSC. This slide explains the procedures to follow, or the steps to follow, when you have actually completed your experimental procedures in a BSC. So we call it BSC shutdown procedure. That means after you finish work, how should you bring out the material, how should you turn off the BSC, etc. So I also strongly suggest you watch the video, this timing or this part of the video, to understand this shutdown procedure. Okay, So that means first thing, when you have finished your work, okay, you should let the air to stabilize first, okay, because you have been moving a lot, okay, and so you allow sufficient time for the air in the BSE to pass through the filters, okay, so that you can actually contain the aerosols and remove the aerosols. Then you have to make sure that all the contaminated waste has been placed into a biohazard bag inside the cabinet. That means, remember, you can keep a biohazard bag in the contaminated area of the BSE. So whatever contaminated waste, you should actually dispose it within the cabinet into the biohazard bag. Okay. Then usually when you're handling biological agents in a biosafety cabinet, you are wearing a double layer of gloves. So you can remove the outer layer of gloves because this can be contaminated with all the infectious agents. Okay, so you can remove the outer layer of gloves and dispose it into the biohazard bag inside the cabinet. Then you can go and put on new outer layer of gloves. Okay, then before you bring out the items from the biosafety cabinet, you also have to disinfect them again. Okay, and including your waste bags. Okay, after you have removed the materials, you again have to surface disinfect the work surface of the BSE using a suitable disinfectant. After you have done that, you can actually close the sash of the BSE. Okay, then you can turn off the light and the blower, and you can actually turn on the UV light to disinfect the work surface. After that, finally, you can actually remove your gloves, dispose them, and wash your hands. When you're handling biological agents, you should also know how to handle biological spills. That means if a spillage occurs from a particular biological agent. Okay. So the biological spill response actually depends on the pathogen's risk group. Remember, I mentioned earlier, right? The biological agents are classified into different risk groups, right? So if it is a risk group 1 organism spill, we consider it as a low risk spill. Okay. If it is risk group 2, it is considered a moderate risk spill. If it is risk group 3 and 4, okay, which are more hazardous, we consider it as a high risk spill. All right. So in this lecture, you are going to learn the steps to clean up a spill if it is risk group 1, or if that means if it is a low risk spill. Okay. However, let's say if it is risk group 2, 3, or 4, the procedure is slightly different. And sometimes, example, if it is high risk spill, we also need trained or qualified people to clean up these spills. Okay, so please remember that. So let's see what you have to do if there's a risk group 1 biological spill. Okay, so the immediate action is you have to notify others. Okay, that means to stop contamination of other people and environment. Then in the event of spill, if you have contaminated clothing, you have to remove them, wash your hands with soap and water. So these are the steps to clean up risk group 1 biological spill. Okay, so first thing, you need to put on appropriate PPE. So for biological spill cleanup, you are required to wear a double layer of disposable gloves and other required PPE. And if there are any broken glass or sharp items, you have to use forceps to pick them up and dispose into a sharps bin. Okay, then you need to lay paper towels or any other absorbent material over the spill and pour or spray any appropriate disinfectant solution. We can use 70% ethanol. Sometimes we have to use bleach solution. Okay, then you have to give it some contact time. So we leave it for at least 10 minutes for the disinfection to occur. Then after that, you can actually clean it up. That means we don't usually use hands, although we are wearing double layer of gloves. You can use a mechanical device like tongs to pick up the material and place it inside the biohazard bag and send to autoclave. Okay, when you're picking up the materials, then finally you have to actually remove the outer layer of your gloves, which may be contaminated with the spill cleanup process. Then you also place it in the biohazard bag. All right. Then after you have sent it to autoclave, okay, you can dispose your remaining gloves into a biohazard bin and wash your hands thoroughly with soap and water. Let's see what are the storage requirements for biological hazards. Okay, generally, because they are biological agents, we store them in fridges, freezers, sometimes in liquid nitrogen tanks, like cell cultures. We store them in liquid nitrogen tanks. The temperature is like minus 196 degrees Celsius. Okay, so when you store biological agents in these units, such as your fridges or freezers, you must make sure that they are properly labeled. Okay, the biological agent's name, etc. Then you also must paste a biohazard sign in these fridges or freezers or liquid nitrogen tanks so that people know that there are biological hazards inside. All right, and because um, they are very sensitive to temperature changes, so in these equipment you have to actually monitor the temperature closely and record it. And sometimes some facilities will actually lock these units. That means not everyone can actually access this because these are biological hazards. Certain biological hazards can be used as bioweapons. So in order to avoid any unauthorized access, uh, better to keep them under lock and key. Lastly, when we are handling biological agents, we should know how to do proper disposal of these hazards. Okay, so there will be waste material generated from your work procedures involving biological agents. So we usually categorize them into either solid biohazardous waste, liquid biohazardous waste, or sharps. Okay, so your solid biohazardous waste can be your contaminated gloves. If you're doing microbiology work, if you have like agar plates with bacterial cultures, you have bacterial or cell culture vials or tubes, disposable pipettes, all these comes under solid biohazardous waste. These must be actually disposed into either biohazard bags or bins, okay, then sent to autoclave prior to disposal. So autoclaving is a sterilization method, okay, that you can kill off any form of live microorganisms, okay. So you need to do this to inactivate them, okay, sterilize them before you actually dispose. So if it is liquid biohazardous, for example, if we have cell culture media or bacterial culture broths, these are liquid biohazardous waste. There are two ways you can actually decontaminate them before disposal. One is by autoclaving, as I mentioned earlier. The other one, method is chemical inactivation. So for chemical inactivation, we can soak the liquid biohazardous waste with certain chemicals like 10% bleach solution. Okay, then you have to dispose them into designated waste containers. Okay, if it is sharp biohazardous waste, that means if you use needles or sharps, okay, which are contaminated with biological material, they must be directly disposed into biohazardous sharps bins, which are located in the labs. These bins should be puncture proof, and you should never recap or clip or try to remove the needle from the disposable syringes, because in the event you may actually get accidental punctures or cuts. Okay, so avoid removing the needles from the syringes or try to recap. uh, Without that, you have to actually uh, place the complete assembly. That means if a needle is attached to a disposable syringe, you dispose the complete assembly into the sharps bin without removing it.
Hello students, I'm going to give an explanation on the comparison of three very important laboratory hoods used in the lab. They are BSC, Biological Safety Cabinet or Bio Safety Cabinet, LFH, Laminar Flow Hood, and CHF, Chemical Film Hoods. They are similar and yet have some differences. So let's start with the first one, Biosafety Cabinet, BSC. So this is the external look of a BSC, and this is the airflow illustration in a BSC. What I want to bring to your attention is that in a typical BSC, this is a class 2 type A2 type, the air is drawn into the BSC from outside. So some of the air is entering the BSC from the ambient environment. right? So the red air and the red color indicate the air is not clean, right? Not clean. While the blue color means it's been filtered and it's considered clean and sterile air. So after the air is joined into the cabinet, it goes downwards into the front grill. Front grill, there's a sucking power here, and then it goes underneath the surface, goes back. So the purple color is actually underneath the surface, and then it's sucked up into the above the BSC space, right? And then go into the HEPA filter, where the harmful particulates are filtered away, right? And some of the air, normally about 30% or so, is actually released into the environment. It's ventilated out, while the other 70%, which is filtered clean air, goes down into the into the biosafety cabinet working space. So therefore, this entire space are filled with vertically, either vertical or horizontal flow. In this case, it's vertical flow of laminar flow air, which is streamlined airflow, which doesn't cross over to different parts, and clean air. So in short, BSC, this feature, this circulating air feature, help ensure a clean environment inside this working space, so as to prevent, number one, cross-contamination of different parts inside the BSC, and secondly, prevent harmful materials such as infectious viruses and bacteria from coming out of the BSC and get into contact with the user. So those, and also the third function of BSC is it help protect the environment, because the air that is released into the environment is being filtered by the HEPA filter. So therefore, you can consider the air going out is clean air. So there is no harmful bacteria that can go to the environment, right? So you can read the notes by yourself, but some of the main features will be HEPA filter and air curtain that helps protect the personnel, because there's no air that coming out, only air goes in. Sample protection, because inside it's a clean environment. And environmental protection, as I mentioned, the air that goes out is being filtered. Right. HEPA filter stands for high efficiency particulate air, which are able to remove 99.97% of particles of more than 0.3 micrometer in size. So it's a very effective filtering system that's able to remove the particulates. Particulates are like dusts, air droplets, right? But they don't remove gases and vapors, because gases and vapors tend to be smaller in molecular size. And there are 4 classes of BSC, 1, 2, 3, 4. Okay, next one, I will move on to laminar flow cabinet. Laminar flow cabinet is typically used in biological experiments such as microbiology testing. So the application is kind of similar to BSC in terms of the materials. It's biological samples. So we don't use laminar flow for chemical experiment. We also don't use BSC for chemical experiment. However, there is a big distinction between LFH and BSC. In fact, there are 2 big distinctions. The first one is the airflow pattern. In BSC, as shown here, it's circulating air. The air, 70% of the air is being circulated, right, inside. While in the case of LFH, laminar flow hood, there's no circulation. Air is going in this direction from top. It's been drawn into the laminar flow hood area, and then it's coming out. It's going out from the gap area under the sash, right? So therefore, laminar flow hood is only suitable for low risk biological samples, right? It's only useful for non-infectious or low risk biological samples, because it doesn't protect the users. It doesn't protect the users. What do we mean by protect the users is that if a hood that protects the users, the air will never be drawn towards the user. Instead, the air should be drawn away from the user. Therefore, there's no possibility of bringing harmful materials towards the user. In the case of laminar flow hood, it doesn't. The air is drawn actually towards the outside, which is towards the user. But it's fine if you are handling low risk or non-infectious materials, because if, for example, you are doing E. coli inoculation, then the air doesn't contain harmful materials, because E. coli is, or at least non-pathogenic E. coli is a risk group 1 bacterial. But laminar flow hood does have a HEPA filter on top. Sometimes it's ALPA filter, which is even better than HEPA filter, right? This is to ensure that the air that goes into the working area is clean. So in other words, in laminar flow hood, the working area is clean, it's free of contamination. Therefore, the sample is protected. That's why we can use for biological testing, because in biological testing, such as microbiological testing, we need to ensure the area is clean, is free of contamination. So therefore, laminar flow hood is suitable for biological experiment that is of low risk. It's also suitable for other things such as QC testing, sterility testing of food, for example, preparation of sterile pharmaceutical products, cell culture experiments, provided the cell culture is low risk, right? Certain electronic systems, right? Because those things all require very clean environment, and they themselves don't have much harm. They don't cause harm to people. Lastly is the chemical fume hood. As the name suggests, it's used for chemical experiments that the chemicals that generates toxic fumes, right? Chemical fume hood, the main feature is that it has a very powerful exhaust on top. It has an exhaust system on top that's able to suck up the air upwards. So there are two configurations. One is known as the ductless. Ductless means there is no duct on top, right? Instead, it relies on filter to filter away the harmful particles, or collector, right? That's able to collect the harmful fumes or gases. Another configuration is a ducted one, where the fumes or gases are carried into the duct. Still, it will have a filter, right? But then after that, it's going to be released into the air after filtering. So, as mentioned, the main function of chemical fume hood is to prevent the toxic fumes or gases from going out of the fume hood and being breathed in by the user, right? So it protects the users from breathing in toxic fumes or gases by containing them inside this fume hood. And because of the sucking power by the exhaust, there's also no accumulation of the toxic fumes or gases, right? So the air direction in chemical fume hood is this way. It's going upwards. It's going upward. It's opposite as in the case of a laminar flow hood. So therefore, since it's going this direction, the user is protected. The user is protected because the air is drawn away from him, but the inside, the working space of chemical fume hood is considered not clean environment. Why not clean environment? Because ambient air is considered not clean air, and it is being joined into the chemical fume hood. Therefore, it's not clean environment. So therefore, chemical fume hood is not meant for experiment that require a clean or sterile environment, such as a biological experiments. So we never use chemical fume hood for microbiological testing, right? It's really only used for chemical experiments that involve toxic fumes or gases. And in terms of environmental protection, chemical fume hood does have a filter or also condensers, traps, or scrubbers to prevent vapors, fumes, and dust from being released in the environment. So it does have some filter, right, or condensers, traps, and scrubbers to prevent the vapors and the fumes from release into the environment, right? Filters is mainly for particulate, bigger particles, while condensers, traps, and scrubbers are used for vapors and fumes. Yeah. So I hope this explanation is useful for you to understand this topic.